The Great Barrier Reef is usually described through the life we can see. Towering coral structures rise from the seafloor, schools of fish move through clear water, sea turtles glide above the reef, and sharks patrol the outer edges. But scientists have now revealed that this enormous ecosystem is also controlled by a hidden world containing hundreds of thousands of viruses and hundreds of bacterial species previously unknown to science. Researchers from the University of Queensland and the Australian Institute of Marine Science collected seawater from 48 reefs across the Great Barrier Reef. Instead of focusing on coral, fish, or other visible animals, they searched for fragments of genetic material floating in the water. The results exposed an invisible ecosystem far more diverse than anyone had documented there before. The team reconstructed more than 800,000 microbial genomes. These included over 360,000 distinct viruses and thousands of bacterial and archaeal genomes. Among the microorganisms they identified were more than 500 bacterial species that had never previously been documented. This does not mean the Great Barrier Reef has suddenly been invaded by hundreds of unknown organisms. Most of them were probably there all along. Scientists simply lacked the technology and computing power needed to detect and reconstruct them. A single drop of seawater can contain DNA from thousands of closely related microorganisms. Separating those fragments is like mixing pieces from thousands of different puzzles into one box, removing the pictures from the lids, and then trying to rebuild every puzzle at the same time. For decades, microbiologists were mainly able to study organisms that could be grown in a laboratory. A sample would be placed in a dish containing nutrients, and researchers would wait to see which bacteria multiplied. But most marine microbes do not grow easily under artificial conditions. They may depend on precise temperatures, unusual nutrients, other microorganisms, or chemical relationships that are difficult to reproduce. That meant scientists were studying only a small fraction of the microbial world. The new project used a method called metagenomics. Instead of attempting to grow each organism, researchers extracted all the genetic material found in a seawater sample. Powerful sequencing machines read long pieces of DNA, and computers searched for overlapping sections that could be assembled into individual genomes. Long-read sequencing made the process much more accurate. Older systems often divided DNA into very short fragments. Reconstructing a genome from those pieces was like assembling a puzzle made entirely from tiny sections of blue sky. Longer sequences provided larger pieces containing more recognizable patterns, making it easier to determine which fragments belonged together. After years of sampling, sequencing, and computation, the researchers created the Great Barrier Reef Microbial Genomes database. It gives scientists a reference library of the microscopic life found across the reef's open waters. The comparison with the human microbiome is useful. The human body is not occupied by human cells alone. Vast communities of bacteria, viruses, fungi, and other microorganisms live on the skin, inside the mouth, and throughout the digestive system. These organisms help digest food, train the immune system, produce useful chemicals, and sometimes contribute to disease. The Great Barrier Reef also has a microbiome. Its microbes do not merely float passively through the water. They recycle nutrients, capture energy, decompose organic material, interact with coral, and form the foundation of marine food chains. Many microscopic algae and bacteria use sunlight to produce organic matter. They absorb carbon dioxide and release oxygen, performing a role similar to plants on land. Tiny animals then consume these microorganisms. Those animals are eaten by larger creatures, transferring energy upward through the ecosystem. The visible reef, therefore, depends on organisms that are too small to see. Some bacteria also transform nitrogen into forms that other organisms can use. Others recycle phosphorus, sulfur, carbon, and iron. Without these chemical processes, essential nutrients would become trapped or unavailable. Coral reefs grow in waters that are often surprisingly low in nutrients, so efficient recycling is one reason such rich ecosystems can exist there at all. Viruses are equally important, even though they are usually described only as threats. Marine viruses infect bacteria, algae, and other microorganisms. When an infected cell breaks apart, its nutrients are released back into the surrounding water. This process can redirect energy through the ecosystem and influence which microbial species become dominant. Viruses can also transfer genes between organisms, changing how microbial communities adapt to temperature, nutrients, or other environmental pressures. With more than 360,000 distinct viruses identified, the new database shows that the reef's viral ecosystem is extraordinarily complex. The discovery comes at a critical time. The Great Barrier Reef is being exposed to repeated marine heat waves, coral bleaching, cyclones, sediment runoff, pollution, and outbreaks of coral-eating crown-of-thorns starfish. Scientists monitor coral cover and visible damage, but by the time a reef changes color or loses large areas of coral, the stress may already be advanced. Microorganisms may provide an earlier warning. When temperature, pollution, or nutrient levels change, microbial communities can respond rapidly. Some species increase while others disappear. The balance between useful and harmful organisms may shift before any obvious damage becomes visible to divers or satellites. This creates the possibility of using the reef microbiome as a biological alarm system. Researchers could compare seawater from a reef with the new reference database and identify unusual changes. A rise in certain microbes might indicate heat stress. Other combinations could reveal sediment, sewage, heavy metals, overfishing, or damage following a storm. It would be similar to using a blood test to detect changes inside the human body before severe symptoms appear. The database could also help scientists define what a healthy reef actually looks like at the microscopic level. Coral reefs are not identical. Water temperature, depth, currents, distance from shore, nutrient levels, and local animals all influence their microbial communities. A healthy outer reef may naturally contain different organisms from a healthy coastal reef. Researchers therefore cannot search for one universal microbial pattern. Instead, they can begin identifying the normal range for different locations and seasons. Future samples can then be compared against those baselines. The study included water from 48 reefs, creating broad coverage, but the Great Barrier Reef is enormous. It stretches for more than 2,000 kilometers along Australia's northeastern coast and includes thousands of individual reefs. The new database is a major foundation rather than a complete record of every organism living there. Microbial communities also change over time. A sample collected during summer may differ from one collected during winter. Conditions before, during, and after a coral bleaching event may produce different patterns. Floodwater from rivers can temporarily introduce sediment, nutrients, pollutants, and microbes from land. Long-term monitoring will therefore be essential. The Australian Institute of Marine Science has studied the Great Barrier Reef for more than 40 years. Adding microbial DNA to traditional surveys could reveal changes that were previously invisible. Scientists currently monitor coral cover, fish populations, water quality, crown-of-thorns starfish, bleaching, disease, and cyclone damage. Microbial analysis adds another layer that may connect many of these observations. For example, a reef may still contain substantial coral cover but show a microbial shift associated with rising stress. Managers could investigate before visible decline begins. Alternatively, the return of certain microbial communities after a disturbance might indicate that the ecosystem is recovering even before coral growth becomes obvious. The database may also help distinguish different sources of damage. Heavy metal pollution, excess nutrients, heat stress, and fishing pressure can each affect microorganisms in different ways. Researchers hope that microbial patterns may eventually function like fingerprints, revealing what type of disturbance a reef is experiencing. That would not replace direct measurements. Scientists would still need temperature sensors, chemical testing, satellite observations, and ecological surveys. But microbial DNA could make monitoring faster and more sensitive. Collecting seawater is also less destructive than removing coral or capturing animals. Researchers can lower sampling bottles into the water, filter the microorganisms, preserve the genetic material, and analyze it later. In the future, autonomous vessels or permanent monitoring stations might collect samples repeatedly across remote reefs. The discovery of more than 500 previously unknown bacterial species also shows how little of Earth's biodiversity has been documented. The Great Barrier Reef is one of the most studied marine environments on the planet, yet a broad genetic survey still uncovered hundreds of bacterial species unknown to science and an extraordinary number of viruses. Many of those organisms may also exist in other tropical seas. They may have remained undiscovered because no comparable database existed or because their DNA was difficult to reconstruct using earlier methods. Future comparisons could reveal which microbes are unique to the Great Barrier Reef and which are distributed across reefs around the world. Some may contain genes with useful biological functions. Marine microorganisms produce chemicals that help them compete, communicate, defend themselves, or survive intense sunlight and changing temperatures. Similar discoveries have previously contributed to medicines, enzymes, and biotechnology. However, the immediate value of the project is ecological rather than commercial. Scientists now possess a baseline from which they can begin observing how the invisible reef responds to a rapidly changing environment. The research also changes the way the Great Barrier Reef should be imagined. A coral reef is not simply a collection of coral colonies surrounded by fish. It is a multi-layered living system extending from large animals down to microscopic cells and viruses. A whale moving through the reef depends on food chains that begin with microbes. Coral growth depends on nutrients recycled by invisible organisms. Water quality is influenced by bacteria breaking down material. Viral infections constantly reshape microbial populations. Even the oxygen in the water is partly produced by organisms that most people will never see. The reef's future may therefore depend not only on protecting coral, but on preserving the conditions that support its entire microbiome. Rising temperatures, pollution, and changes in water chemistry may disturb these communities in ways scientists are only beginning to understand. The new map cannot prevent bleaching or stop marine heat waves, but it gives researchers a powerful new way to observe the reef before damage becomes obvious. By learning which microorganisms belong in a healthy ecosystem, scientists may recognize the earliest signs that something is changing. For centuries, the Great Barrier Reef appeared to be built entirely from visible life. Now, genetic sequencing has exposed another reef layered beneath it, one made from bacteria, archaea, microscopic algae, and hundreds of thousands of viruses. This invisible ecosystem has always been there, producing oxygen, recycling nutrients, feeding animals, and quietly supporting one of the greatest concentrations of life on Earth. Scientists have finally begun to map it, and the smallest inhabitants of the Great Barrier Reef may become some of the most important tools for protecting its future.